The overall goal of this procedure is to assess early host-pathogen interactions and the invasion potential of Entamoeba histolytica. First, make preparations of the intact trophozoites, the trophozoite total lysates, or the secreted trophozoite products. Then incubate host epithelial cells with appropriate concentrations of the different trophozoite products. Now, using immunofluorescence, analyze the treated epithelial cultures for select Entamoeba histolytica proteins that interact with tight junctions of the epithelial cells. Also measure alterations of the paracellular barrier by transepithelial electrical resistance. Taken together, results can demonstrate tight junction disassembly by Entamoeba histolytica and also pinpoint the participation of the select complexes in this process.
The key advantages of this technique are the use of different products of the parasite to evaluate the translocation of Entamoeba histolytica factors into the host epithelium, as well as to demonstrate the participation of these Entamoeba histolytica factors on tight junction opening of host cells at very early times of infection.
This method can help answer key questions in the amoeba investigative field, such as understanding the molecular interactions among epithelia and the Entamoeba histolytica parasite.
Generally, individuals new to this method will struggle because they do not know the appropriate incubation times, proportion of the two cell types for co-incubation, and also how to obtain the different histolytica products and how to measure TER.
The implications of this technique